Let's go over the monomer of DNA, which are known as nucleotides. Remember that DNA is our genetic code, and it's going to be really important because it codes for our proteins or our physical characteristics. And the monomer or the building block of DNA is a nucleotide. Now, a nucleotide is made of three different parts. The first part's the phosphate group, the second part is a sugar molecule known as a deoxyribose, and the last part is a nitrogen-containing base, which can be a single or double ring. In the four nucleotides that make up DNA, the phosphate group and the sugar are always going to be the same. However, each of the four different nucleotides is going to have a different nitrogen base. And the four different nucleotides are adenine, guanine, thymine, and cytosine. You can see in this illustration that the phosphate groups and sugars are always the same. However, the bases up here are different. This is because there are two different types of bases. We have pyrimidines and purines. Pyrimidines are single ring nucleotides, and they are cytosine and thymine. Purines are double ring nucleotides, and they are adenine and guanine. So you can see in our purines, 2 rings in guanine and 2 rings in adenine, and in our pyrimidines, only 1 ring, cytosine and thymine. Now, it may look like the purines are exactly the same because of that last picture. However, if we look at the molecular structure between adenine and guanine, you can see that there are differences within the chemicals and the arrangement of the atoms. This just basically means they are different at the molecular level. And the same goes for cytosine and thymine in the pyrimidines. If you look at the chemical structure of each of these bases, you can see that they have different atoms and arrangement of atoms. So even though the 2 purines and the 2 pyrimidines might look identical, they do have molecular structures that are different. And this is going to lead them to only bind or bond to a specific nucleotide. And this is called base pairing. This is when each purine only is going to bind with a specific pyrimidine. So adenine is only going to bind with thymine, and guanine is only going to bind with cytosine. Now you can see there are 2 bonds between A and T and 3 bonds between G and C. This is because there will be 2 hydrogen bonds between adenine and thymine and 3 hydrogen bonds between cytosine and guanine.